So in 8.2, we're going to build on the ideas that we already met in 2.8, and that's looking at cell respiration. So go back to those notes and check in before you carry on here. But the essential idea here is that respiration is a chemical reaction, and it's all about the controlled release of energy from organic compounds, and the controlled release of energy will be in the form of producing ATP. So ATP is this energy currency, and we met this idea already. The syllabus statements are listed here and here, and in the booklet there are a set of questions which are associated with these, which I'd like you to be working through. Recapping then that we already know that this definition of respiration is important. Control, because of enzymes and organic compounds, we can actually release and produce ATP from both fatty acids as well as carbohydrate. Glucose is usually the starting substrate, however. So it's the controlled release of energy to form ATP. This chemical reaction here is an overview, but it's much too simplistic. And actually, as we've already met, this overview is broken down into a number of phases. So ATP, the energy currency, remember, is all about this phosphodiester bond, which is here. So we've got 3 phosphates, and if we break this terminal bond, that is going to allow us to release a lot of energy that can do work. So by producing ATP, we fill up our batteries, and then if we break off that to produce ADP, an inorganic phosphate, remember this is referred to as Pi, not just P, then by breaking off that bond, we can release energy. And what respiration is all about is taking that phosphate and popping it back on there to make ATP. Remember that I've already said to you before that you can't stockpile ATP, so the rate at which ATP is produced is equal to the rate at which it's used up. As we've met in 2.8, we've got a series of reactions, the first of which is glycolysis, then we've got a link reaction if oxygen is available, followed by Krebs cycle, and then we've got this part here, which is the electron transport chain and chemiosmosis, and both of these together are referred to as a process called oxidative phosphorylation, and this is where the ATP actually is produced. So oxidative phosphorylation is both of these together, and in the latter part of this teaching unit, we're going to look at each one of these processes. They are interconnected, but you need to talk about them and differentiate between them very, very clearly. We've also met the idea that if we're doing aerobic respiration takes place in the mitochondria, and we know about the structure of the mitochondria, this double membrane. Connect this to the ideas that we also met about the origin of the mitochondria and endosymbiosis. But what we need to appreciate is that the first step happens out here in the cytosol, but chemiosmosis and the electron transport chain happen in here inside the mitochondria, and this is where most of the ATP is produced. So the mitochondria is sometimes called the powerhouse of the cell. And this question here, what type of cell would have lots of mitochondria? Cells which have got a very, very high energetic demand, so liver cells, muscle cells, etc., etc. Really important that you can draw and label the different components of the mitochondria. So we've got the outer membrane, then we've got this folded inner membrane, and the folded inner membrane folds these projections which are referred to as cristae. The cristae have got an incredible number of proteins embedded in them, and the cristae is extremely important in the process of oxidative phosphorylation, which we come back to later on. Also worth noting is that the gap between the outer and the inner mitochondrial membrane is very, very small. So the volume in here is extremely small, and that also becomes important in the process of chemiosmosis, because what we want to do is put a high concentration of hydrogen ions within this gap. We'll come back to this later on. Also remember that we've got 70 S ribosomes, and we've also got naked DNA, all of which are indicators which support the idea, the notion of endosymbiosis. It's a small organelle as well, as you can see here with the scale bar. So this is an overview which allows us to have a look in 3 dimensions. So these cristae are really important, and the inner mitochondrial membrane, the space here, is extremely important later on. here in the matrix, we've got many enzymes which are allowing us to do lots of chemical reactions inside here too. So this idea of structure function, which we come back to at the end, is super important. Here you can see an electron micrograph, and it's important again to be able to recognize this as a mitochondria and be able to add labels on if you were asked to do this in an exam. Also use a scale bar to calculate magnification, etc., so links and connections to earlier parts of the course. What we've got here are some really good links. Please make sure that you go back and have a look at this. But the idea here is that this technique called electron tomography is how we know about the shape and the dimensions and the structures within the mitochondria. So technology, again, allowing us to see the unseen. We need to make sure we can define and differentiate between the idea of reduction and oxidation. And we've got this nomenclature of oil rig, which might be helpful for you to appreciate. So we did this when we did 2.8, and hopefully everybody is clear. If you're not, you must make sure that you ask me about this. This is particularly important when we talk about glycolysis and also when we talk about Krebs cycle. So oxidation is the loss of electrons or the loss of hydrogen ions, and that's really important. So when we do oxidation in glycolysis, we're taking hydrogen away from the sugar, so we're oxidizing the sugar. And remember, what we do is we give those hydrogen atoms to a carrier molecule, NAD and FAD. So when we take away the hydrogen from the sugar, we're oxidizing the sugar. When we give that hydrogen to NAD, we are reducing the NAD. Here you can see this as a chemical reaction. So we've got this electron carrier or cofactor, and remember that this is reversible. We can reduce it by adding hydrogen. The hydrogen comes from the sugar. And later on, when we talk about oxidative phosphorylation, we're going to talk about taking that hydrogen back and then reoxidizing that back to NAD. Got another carrier, which is called FAD, which is important in Krebs cycle. So let's look at these step by step, and let's start with glycolysis. And glycolysis, you've got to make sure that you appreciate what each of these chemical reactions is all about. So phosphorylation is all about moving a phosphate group from one molecule to another molecule. If we're moving it from one molecule to another molecule, this kind of phosphorylation is called substrate level phosphorylation. What we do at the beginning is we phosphorylaze the sugar, and we add 2 phosphate groups to the sugar to make it really unstable, which allows us then to do the next step. At the end of the process, what we do is we take the phosphate from the sugar and we pop it back onto ADP, so we form ATP down here. But what we're doing in both cases is substrate level phosphorylation. The second step is lysis, and we take our 6-carbon sugar with the 2 phosphate groups on, so which is called hexose biphosphate, and that 6-carbon sugar is very unstable and will readily split into 2 3-carbon sugars, so we're going to produce triose phosphate. What we then do is oxidize the sugar, and we take hydrogen from the sugar and we're going to give it to a cofactor NAD, so we reduce the NAD. We're doing an oxidation process and a reduction process at the same time. This comes from BioNinja. BioNinja has got some really good images, I think, and I've taken a few and added them into this PowerPoint, but I would strongly urge you to look through each of the pages which are posted on BioNinja. But here we've got the overview, so we're going to start usually this process with a 6-carbon sugar, which is normally glucose. Then what we're going to do is we're going to use 2 molecules of ATP. We're going to take the phosphate from the ATP and we're going to add it to the glucose. So we're doing substrate level phosphorylation. We're making the glucose unstable. We're forming that unstable 6-carbon sugar, then is going to break, it's going to do lysis, and it's going to break into 2 lots of a 3-carbon sugar, each of which has a phosphate group on it. Then what we're going to do is we're going to take the hydrogen from the sugar. So we're going to oxidize the sugar here, and we're going to give it to NAD to produce reduced NAD, or NADH plus H+. Then what we do again is substrate level phosphorylation to produce ATP, and ultimately we make 2 molecules of pyruvate. Remember, we went through this in 2.8, but this is a nice overview, but must make sure that you can use the language of the reactions that are occurring here. go into the Google Classroom, find these links, play these links, just make sure that this is all clear in your head. Very helpful to support this. And here again, we've got the same sort of summary, but here each one of these yellow circles is meant to represent a carbon. So we've got a 6-carbon sugar here, which is a hexose sugar. We're going to phosphorylate it by taking ATP, breaking that bond and adding the phosphate group onto our sugar. So here we've got biphosphate. That biphosphate, because it's unstable, undergoes lysis. So we're going to produce 2 of these triose phosphates. And then we're going to undergo oxidation of the sugar and reduction of NAD. So we're going to produce NADH plus H plus. More substrate level phosphorylation to take the phosphate from these sugars and pass it back to ADP to form ATP. And the final product here is 2 molecules of pyruvate. So overall, we're going to use up 2 molecules of ATP. We're actually going to produce 4. At this stage here, there's also something else going on, which is not part of your specifications, but we've got another phosphate actually moving to a molecule here, moving away from a molecule, which is allowing those 4 to be produced. And what we've got is a net yield, therefore, of 2 molecules of ATP, 2 molecules of NADH+, and 2 molecules of pyruvate per sugar molecule. The enzyme that's allowing us, a very important enzyme that's doing the first process, is called hexokinase. And hexokinase is responsible for adding those phosphate groups. And as we've said already, the phosphorylation process makes the sugar much more unstable, and that allows us to proceed with the reaction. And there we go, we have the summary for you. The next step is then to look at a reminder that we have to decide now whether we're doing aerobic or anaerobic respiration. If we do aerobic respiration, the next step after glycolysis is going to be the link reaction followed by oxidative phosphorylation. But we can only do the link reaction, Krebs cycle, and oxidative phosphorylation if oxygen is available. If oxygen is not available, instead what we need to do is go down the anaerobic route, which we talked about in 2.8. So make sure that you're clear with this. And remember that what we're doing here is we're taking the hydrogen off the NAD that we produced in glycolysis, and we're adding it onto the pyruvate to produce lactate in the case of animals. What we also do is add it to the pyruvate and then decarboxylase if you are a plant or a bacteria to produce ethanol. So this ethanol is a 2-carbon sugar and the other carbon is here. But what we're doing is if oxygen is not available, we need to re-oxidize this NAD so the NAD can still carry on doing glycolysis and still produce small quantities of ATP. But go back to the notes that you've got from 2.8 and make sure that this is clear. But in 8.2, what you need to appreciate is that we're looking at aerobic respiration. So what we do is we take the pyruvate and instead we're going to do the link reaction followed by Krebs cycle, followed by oxidative phosphorylation, which is the electron transport chain and chemiosmosis combined. So that will take us to video 2.